first question. Okay. A. Yes. Yes. This part is from the human reproductive system, and especially this is from female reproductive system. So fallopian tube, the main function of fallopian tube when in the ovaries the egg will be produced, ovum will be produced. It helps to transfer it. The fimbriary region will help to accept that ovum, and then it passes through the main fallopian tube for if fertilization may happen. So the main, if we talk about, this is about the movement, the thing. So if this is a movement, then epithelial cell with the cilia, means cilia helps for the movement. So if we put this logic, then it will be easy for us to identify what will be the correct option from this. So ciliated epithelia will help to the movement of the cilia from the epithelial cells will help for the movement of the ovum in the fallopian tube and then ultimately to the uterus. So option A is the correct one. Next. Anyone? C. Okay. Okay. So this question is from plant kingdom part of the plant classification. So the varieties that we are getting is there are two types of the reproductive systems. One is the asexual where the gametes are not involved, and then is a sexual kind of reproduction where gametes are involved. So if though you don't know the detailed structure at that means if you haven't gone through, you can logify this statement very easily. We have to point not a sexual kind of a reproduction. So that is the something which is dealing with the sexual one. We have to choose those options. So if you see the options carefully, usually spores are the methods of the sexual reproduction. So you can definitely remove option C. Okay. Then the word gametes is associated with the sexual reproduction kind of a thing. So different kinds of. So though you don't know the meaning of the isogametes, you can definitely put your logic that it is the something related with the gametes, and that is one with the definitely related with the sexual kind of a reproduction. And we have to select such option which is not a sexual kind of a reproduction. So by logic, you can select option A, which is the correct one. While the other options, the conidia of the penicillium, the zoospores of the chlamydomonas, and gamules of the sponge are all kinds of the asexual reproduction. These are the structure which are associated with the asexual reproduction. So if you don't, at least the conceptual understanding will help you to select the option at that time. Okay. So for this question, option A is the correct one. Next. Yes, option A is the correct option because palindrome is such sequences which are the sites for usually for the restriction enzyme, such sequences. One thing. So they are usually reading, if you read the sequence in the both direction, they will be the exactly same thing. So if this is case only in case of the option A because GAATTC, and if you read in the reverse direction, it will again give you the same sequence GAATTC, which is the example of the palindrome. So option A is the correct one. Next. Option, okay. Yes, option A is the correct one. IgE type of antibodies are associated with the allergic kind of a reaction, especially for the pollen content. This part is from the microbes and diseases, especially the subsection of the immunity. So this is one of the if you now go through, now this is our fifth paper that we are referring to, and definitely at least one question is associated with the immunological perspective. So I definitely recommend you all to go through that section. And this kind of a charts will help you to remember things in a better way. So five types of antibodies are there. There are different. They are in different shapes. We can see. So IgA is associated. It's secreted into the mucus, saliva, and tears, and usually it is associated with the pathogen destruction. IgD is a B cell receptor, especially mast cells and basophils, and um, it stimulates for release of the IgM type of antibodies. IgE binds to the mast cells and basophils. Sorry, that is a characteristic of IgE type of antibody, not IgD. And it is associated with the allergic kind of a, or antiparasitic kind of a response. IgG binds to the phagocytes and main blood antibody. So if you can see in this particular chart, the percentage of total antibodies in serum, the IgG is the highest percentage in the blood serum that we have, and IgE is the least one. So this type of a charts will help you to remember things in a better way. So any question, so you have to remember the functionality, what is they are really associated with. So it helps you to answer it quite well, and the percentile part especially, which is the most abundant one in the serum, so that will help you. Or even what will be the crosses the placenta, means at the time of placental transformation, if you can see, only IgG can pass through the mother to the baby. So it's a placental transformation is happening for IgG type of antibody. So this type of a charts will help you to remember things in a better way. So for this question, option A is the correct one. Next. Option A. Yes, option A is the correct one. Reduction in population size. So this is associated with evolution, and Hardy-Weinberg is one of the favorite chapters for IISER Aptitude test. So you can remember we want to find the Hardy-Weinberg, means the allelic frequency is in the equilibrium. That's what means recessive and dominant characteristics. Whatever allelic frequency have, that is usually in equilibrium. But there are various factors which can deviate the means the equilibrium can be disturbed. This because of the mutations that can happen, gene flow that is happening in the given population. Genetic drift means the chance event that can happen, which can change the allelic frequency at large. That is what the genetic drift. Amongst this, the reduction in population size, though the population size is reduced, there is a least chance of the deviation that can disturb the Hardy-Weinberg equilibrium. So option A is the correct one. Next. Option B. Okay. Yes, I can hear you. Option B. Okay. Okay. Sure, sure. A or D. Option A. Okay, okay, okay. So this question is from molecular basis of inheritance. So this is a classic example when people don't know that DNA is a substance which is carrying the information for the different types of a character. So the Frederick Griffiths experiment with Streptococcus pneumoniae. Two strains of Streptococcus pneumoniae he studied. One is a smooth strain, means the apparent colony is smooth because of the different kind of protein is secreted, and which is have the virulent characteristic, means that kind of a strain can able to cause the pneumonial diseases. And another is a R, which is a rough kind of a strain, which is a non-shiny, you can say, the rough one in the appearance. And those are the one which are virulent, so that they are not infectious one. So if they are injected, the rough colonies will be injected, then the mice will be alive. And if the smooth colonies will be injected, the mice will be dead. So we can have the answer for T5 and T6. So this smooth strain will be die, this will be die, and this will be alive. Now the other cases. The yes, Anagha. Answer is D. Okay. So let's see each cases. So if this is basic understanding that we have the smooth strain, the S strain, if injected in the mice, it will cause the pneumonia and ultimately mice will die. And if only rough strain, which is alive strain, is injected in mice, the mice will be alive. So it won't die. So by understanding this, let's see the first case, that is the T1. So the S strain is heat killed, and it is injected in mice. If it is heat killed, then the mice will remain alive, because already the strain is heat killed. Because of that, the mice remain alive. And this is a classic experiment which is done by Frederick Griffith earlier also. In his experiment also, he got this kind of a disease, this kind of a inference, sorry. In case of the T2, the heat strain, which is a heat kill, and R strain, which is a live, is togetherly injected in mice. At that time, Griffith even observed that the mice is eventually dying. This is because the DNA present in the heat from the heat kill strain, when come in contact with the live system, it is get transferred into the live system, and that makes the R strain more virulent, and ultimately the mice will die. So for this particular thing, for smooth strain, when injected in the in the heat kill form, the mice will live, that is the alive. And if this is the case, if the S strain, that is heat killed, is injected into the in association with the R strain, that is the live strain, is injected, then the mice will ultimately die. So this is a dead condition. Now the third condition, that is the T3. T3, okay. T3 is the condition where the nucleic acid isolated from the S strain. So live strain, which can cause the virulence, have characteristic is heat kill in nature, plus R strain from the live one, and it is incubated with the RNAs and injected in mice. If this is the case, the nucleic acid is DNA in nature. So RNAs have no activity on that particular thing. So that means the DNA will be still have the virulence characteristic, and it is injected in the R strain. So then it can again become the virulent, and the mice will ultimately die. So this is again death kind of a situation. And T4, the nucleic acid isolated from the S strain plus R strain is a live strain, and it is incubated with the DNAs. DNAs can able to degrade the DNA. External DNA is there, and if DNase treatment is given, that can shear the DNA. So that means activity of this nucleic acid will diminish because of the DNase activity, and only R strain will be remaining. R strain is a virulent in nature. So that means the mice will be live. So if we have to find this particular pattern, first will be the live, then dead, dead, live, dead, and live. This is in case of the option A. So option A is the correct one. For this particular question, you need to remember basic characteristic that we have. The S strain is a virulent one, means disease causing one, and R strain is a virulent one. If heat kill situation is there, the classic example is already there. And again, the two extra cases that they have put forth is T3 and T4. The RNAs will not have any effect on the DNA, but DNAs have effect on the DNA, which can shear it. So that means if the virulent DNA is not there, if the only strain is the virulent one, that the mice will be ultimately will be alive in nature. That is what kind of search is then done by the Avery, MacLeod and McCarthy kind of And then they discovered ultimately that yes, as the DNA's activity is only affecting the presence of that particular virulent characteristic, which is transforming factor, that is ultimately nothing but DNA. So option A will be the correct one. Next. B, okay. B, option B, okay. Okay, so this is from the chapter of the photosynthesis. So we have already aware that two types of reaction. One is a non-cyclic kind of photophosphorylation, and another is a just cyclic photophosphorylation. So see, I'm purposefully today underlining the words. So those are the keywords which you must consider when you are reading the question, what they have asked. So we are talking about photosynthesis, the products generated from them, and the main keyword in this question is a far red light. So if you see the red light spectrum is quite large one. Okay, so even 680 falls in the red light spectrum and 700 is also falling in the red light spectrum. However, when we talk about the far red light uh, wavelength is fall only from 700 to 800. That range is for the far red light. So if the plant is exposed to only far red light, at that time only PS1 will be activated. So only PS1 can be more functional because that have the capacity to absorb the wavelength of the 700 nanometers. So if this is a case, if we are considering this particular case, if only PS1 is involved in this process, that is associated with the cyclic photophosphorylation. And in the cyclic photophosphorylation, only ATP generation will be happening because no NADPH producing enzymes are there at that time, and even not water splitting will be happening. If the question is about just a red light, then we can say the two systems will be worked out. That is PS2, which have the absorption maxima at 680, and PS1 at the 700. At that time, in non-cyclic photophosphorylation, three products are formed. One is the ATP, another is NADPH, and third is the oxygen. That is the case that is the product formed during the non cyclic photophosphorylation, but at that time two red zones will be. Here we are referring only to the far red light, so only PS1 will be involved, and that will be only for the generation of the ATP. So option A will be the correct one. Somebody has raised hand, right? Shreyas? Yeah, okay, okay, Roshan. Yes, yes, option A is the yes, yes, that is correct one. Next. Option A. Okay. Anyone else? Sure. A. So this is from the biotechnology part, biotechnology principles and processes. So we have to cut the restriction. Restriction enzyme sites are very specific. So this is total 400 base pair plasmid. So it's all in all, we have to see the cut. So firstly, what they have done, they have the cut with the EST1 and then with the hint 2. So at that time, two fragments will be formed ultimately. And then again with the ECOR1 and SAL1. So ultimately we will get four fragments, 1, 2, 3 and the remaining one. So for if we determine the sizes of all these three, we can determine the remaining size. So 1700, this is the position. So 1700 minus 800, that will give us 900 kind of a base pair. First fragment will be like this. So 900 is only option present in option A. So you can easily select. Of course, you can re-verify with the remaining fragments also. But however, this can help you to shortlist in the earliest. Then 1920 minus 1700, that will gives us 220, and 2460 minus 1920, that will gives us 540. So ultimately if we sum up this all, then we will get 400 base pair of the plasmid, and which is the correct answer. So just this is a very simple case. Actually they have detailed, there are no multiple sites so of the same restriction enzyme. So whenever this kind of a pattern is another one of the favorite pattern for IISER aptitude. So just before you are doing everything, just make sure that the remaining pattern means the ultimately whatever happening in the whole scenario, there are no multiple sites of the same enzyme. Please make sure. Otherwise, for example, if there is one more PST1 site is present in different location, the size of the pattern will change. So sometimes they will give you gel electrophoresis image, and just they will just give you the image, and then you have to just which image can be formed based on the different restriction enzyme sites. So based on that, we have to choose answer accordingly. Yes, yes, Anudha. For example, yeah, obviously, but number of fragments will change. Ultimately, you will get in all total will be the 400 base pair. That is by addition you can see. Means for example, let's imagine here will be one more PST1 site. So if the first cutting will be done at that time, PST1 will cut here and here also. So ultimately once the SAL1 will be get cut, the things will be little bit different. The sizes will be changed because right now we are considering the remaining portion as this much. However, if PST1 additional size will be there, then one more cut will be there. So ultimately five fragments will be formed. Like so, that's what I'm saying. Here just, this is a very simple case, only single site. I'm just giving you one more insight if multiple sites are present, and if instead of giving the size, they will just give the gel electrophoresis image that they will have three fragments, four fragments, and you have to choose the correct gel electrophoresis pattern. Then you have to choose very carefully if they have the multiple sites. Okay? Yeah. Next question. Option A. Okay. Yes, this is regarding the photosynthesis pattern of the two classic cases that we have discussed is about the C4 plant and C3 plants. So C3 plants, the crepe, the ultimately the photosynthesis will be happening in almost all kinds of plants. That is, crepe cycle will happening in everywhere. However, it is the differentiation about the mesophilic tissues and the vascular bundle sheet that we have discussed. So the C4 plants characteristic, they are growing in the high temperature zones. This is the one main characteristic. So if we are talking about the leaf temperature, if we see P2 curve, it at the higher level, and the rate of the photosynthesis is very high in case of the C4 plants. So their leaf temperature is also very high, and they can present in the tropical adaptation. They have the better for the tropical adaptation because the high sun rays are saturated and the intensity is also very high. And the rate of the photosynthesis is ultimately very high for the C4 plants. In case of the C3 plants, their leaf temperature sensitivity is also falls in very narrow range, and the photosynthesis rate of the photosynthesis is lesser than the C4 plants. So if that basic concept is clear for us, then it is very easy to choose the answer. So option A is the correct one. Next. Option C. Okay. Anyone? A. Okay. So this particular question is from the first chapter of the ecological unit. This is from the organisms and populations. So two maps are given. We have to correlate the relation between them. First is a external temperature and internal body temperature, means how organisms are reacting with each other. And secondly, body size and the surface area, that is the volume that they have. So, if you remember the first part, the different characteristics which can affect the organism's behavior is that two kinds of organisms are there. One, when the external temperature will be changed, they don't want to change their inner temperature. So, they try various methods to maintain their body temperatures. For example, in case of mammals, when there is a summer, we sweat to maintain. In the cold, we shiver to get more. This is a heat kind of generating example that we have seen. So if this, yes, Roshan, okay, okay, okay. So if this is the case, then we have to see two things. External temperature maintaining, if whatever external temperature is there, if the organisms which are maintaining their inner temperature irrespective of that, such organisms we called as a regulators. So x-axis will be representing regulators. And the another class of the examples, so main examples of the regulators are the birds and mammals, are the main example from the regulator kind of a perspective. Second, there are conformers. So conformers are the one when the external environment change, they will make change in their body temperature to adapt in a better way. So that is the means except birds and mammals, majority of the organisms fall into this particular category. So if you can see as the external environment the temperature gets rising, their internal body temperatures is also increases. So this is a case of the conformer. This is the one logic that you can use. So you have to see the regulators one first. So if we are talking about the mammals and birds, we can find the animals that we have to find the relation is the hummingbird and polar bear. So these are the two categories that you can definitely consider under the regulators, and crocodile and frog will be under the conformers. Now the second kind means second group that we are referring to. So we have seen that in that particular chapter, the smaller the size, the area of the surface area and the volume ratio is higher in case of the smaller kinds of animals. So that is the case why in the polar region we don't find any much smaller kind of animals, because they are difficult to adapt them to get that kind of a condition. So if this is the case, we can see the group A will representing as a smaller, much smaller animals, and group B will be representing the surface and volume area which have the lesser, which are the bigger animals. So in this particular group, we can see crocodile and polar bears are the bigger ones. So we can consider them for the B, and the frog and hummingbirds are the smaller one with the smaller surface area and the volume ratio. According to that, they will form under the A group. So if we want to now understand this concept, we can match the thing. So let's see each option. So XA, that is the hummingbird case that we are discussing. So hummingbird X, yes, it's a regulator kind of a thing, and A, it falls in the A group. That is the surface area wise, it is correct. Then Y, next is a crocodile, which is a Y, which is a conformer kind of a thing, and which has a bigger surface area, means it's a large kind of animal. So XB is the correct one. Then the frog. Frog is again the conformer, so that is following the Y kind of a relationship, and it is also the smaller one, so it will be A kind of a relationship in the second part. So Y A is also correct. And then the polar bear, which is a again the regulator kind of a organism, and then it is a larger surface area. So XB is again correct. So all the things are correlated with the option A. So option A will be the correct one. Next. Option A. Okay. So this is one of the question which is, again, you can put the logic over here if you don't know the exact answer. So we are talking about nitrogen cycle because many process means it is a mixed kind of a data that they have given which can find interesting. So we have talked about the nitrogen cycles. So we have the two processes, so ammonification and nitrification, the processes that will be involved for conversion of atmospheric nitrogen. So when ammonia is formed and it requires photons from that is the H plus ions from the photonic energy, then it can be ultimately forms the protonation of the ammonium ions will happen. This is the basic process which is described in first part. Then there are various enzymatic systems which are making the convergence. So we have to ultimately find the conversion of the alpha keto acid which is into the amino acid. So if you are seeing the options over here, the glutamate dehydrogenase will be ultimately converted into acidic form, which is a glutamic acid. And glutamic acid is one of the name of the one of the example of the amino acids. So you can easily correlate this by the logical way if you are not really sure about the glutamic dehydrogenase or not. Nitrogenase is for the enzymatic, it's for the fixation of the means for the ammonification process, the nitrogenase is required. So that is not the case for the uh, making of the amino acid synthesis. Trans-acetyl is definitely not a option, and lactate dehydrogenase again it is not playing role in the ammonification or nitrogen fixation process in that to ultimately form the amino acid synthesis. So you can by putting the logic, if you don't know the exact answer, as I said earlier also, two options are there. If you know exact answer, that is well and good. You can choose the option very easily. At least if you don't know the options, then at that time you have to use the strategy. If you don't know, definitely means I can easily eliminate trans-acetylase and lactate dehydrogenase very easily. If this is the case for you, try to eliminate the maximum which are unlikely scenario, which will lead you to ultimately the one which can be the selected. So you can use the same logic for solving some questions where you don't know exact answer. So for this question, option A is the correct one. Next. Option A, okay. Option A. So this is a combined question actually. This is in the digestive system part and from uh, Roshan, if you can unmute yourself, it is just disturbing. Uh, so this is a combination question. So one is from the digestive system, which is part actually excluded from this year's. Means if you are referring 23-24 kind of a syllabus, they have actually discontinued the digestive system part. Still, we can find it in 21 question, and the another is excretory system, which is still in the function. So if you know the function of the small intestine is the last part where the ultimate digestion means complete digestion process will be happening. So all complex substances will be ultimately divided, formed into the simpler form. Means fats will be converted into fatty acid. Then the carbohydrates will be converted into glucose, and protons are converted. Means they will be in the amino acids. So the complete conversion and absorption will be happening in case of the small intestine. So, okay, 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 okay. So, small intestine is doing the complete absorption of the nutrients in the ultimate form. Renal tubules, when we say renal tubules for the particular structure will be divided into the Bowman's capsule, then proximal convolutory tubule, distal convolutory tubule, and the ultimate form the convolutory duct or the convolutory tubules. However, convolutory tubules are usually not considered part of the ultimate renal tubules because it is the many nephrons are come into the one ultimately into the one many nephron units are get associated with one collecting duct where from which the excretory system will be part. So if this is the case, the mainly function of the small intestine is absorption. Absorption of the nutrient is the in the smallest form is the one thing that we are related with. And the renal tubules is also associated with the maximum absorption of the glucose, which will be taking place in Bowman's capsule and also in PCT. So if we see the options of the absorption of the glucose, yes, we can find both in the small intestine and in the renal tubules. Excretion of the waste material, which is not happening in case of the small intestine, but happening in case of the renal tubules, but only happening at the time of the collecting duct. That is the last part will be happening. So that is not the case. Excretion of the water, which is not happening in case of the small intestine, and absorption of the protein is not the function of the renal tubules. The absorption of the proteins in the smallest amino acid form is happening in case of the small intestine, but not happening in case of the renal tubules. It is the only glucose uptake which or absorption of glucose which is a common function between the small intestine and renal tubules. So option A will be the correct one. Next. Yes. So coil bird. Option A. Okay. Cuckoo is coil bird. Option A. So this type of a match means this is from the biodiversity part, and we talked about different kinds of correlations that we have, the parasitic, commensalism, mutualism. So I hope you remember the conceptual understanding of each. Match the following is I see little bit easy kind of a questions, because if you know at least two of them means for sure if you are, and then you can choose from the answers where the two are falling exactly, and then you can negate the others at that time. So firstly, the herbivores and plants. Amongst the option, this is kind of a relation of the predation, because prey-predator kind of a relationship. We don't have to always imagine prey and predator is always kind of a carnivorous kind of a relationship. Here the herbivores can feeding on the plants. So this is a prey-predator kind of a relationship. So option B is the correct one. Then cuckoo and crow is the example of the parasitism. This is a classic case of parasitism that we have thought that the cuckoo birds are lay their eggs in the crow's nest and remove the crow's own eggs at that time, and the birds even is more parasitic. They are this is a parasitic kind of a relationship. This is a classic example that we have discussed in the biodiversity, in the ecological balance in that particular part. Then sea anemone and the clownfish is an example of the commensalism. So here the sea anemone and clownfish are in association with each other, where the sea anemone get advantages from the clownfish. However, clownfish is not getting either affected or not get um, harmed either or. So this is a commensal kind of a relationship. And fungus and cyanobacteria, this is lichen is a classic case of the mutualism. So if you at least see the option A and D, then based on the pairs, you can select the option A will be the correct one for this question. Next. Option A. Okay. So from the cell division part is another favorite chapter for IISER Aptitude, and amongst them the meiosis is because it's much more complex and many things we have to remember for this particular thing. This is again the one of the part for interest for the as per your exam is concerned. So meiosis, if you see the options they have given, the appearance of the recombinant nodules, the formation of meiotic spindle, formation of the chiasmata, and synaptonomial complex. We have to firstly consider the prophase one, which is the most complex one, and many things happening in that particular part. So leptotin, zygotin, pachytin, diplotin, and dikinesis. These are the 5 phases that and what is happening at each process. So firstly, in leptotin, the firstly the condensation will start for the both different kinds of chromosomes will be so this is the condensation part. Then in the zygotin, the synaptonomial complex will be first formed, means the pairing will be start and synapse formation will be happening in this particular case. If this is the case, then option 4 we have to consider. This is the first part. This is what is happening in zygotin. Then in pachytin, the crossing over event will be started. So that means the non-sister chromatid will overlap and the recombinant nodule will be appearing. So at that time, no sharing is already done. However, that complex formation is already starting, means that crossing over event will be happening in pachytin. So this will be happening after that. So leptotin, zygotin, pachytin, diplotin. What is happening? The complete sharing with the non-sister chromatid will be happening, which is a recombination event, and that particular sharing will be called as a formation of the chiasmata. It is the chiasma formation, which is the next case in the case of the diplotin. And in case of the dikinesis, the complete sharing is completed, and the next will be the metaphase 1. Then the mitotic spindle formation will be happening in dikinesis, which will be ultimately start to arrange at the time of the metaphase. So that will be case 2. So if this is the arrangement that we are talking, 4, 1, 3 and 2. So option A will be the correct one. So please just impregnate your mind that first prophase 1 will be the most interesting part to ask question about. So if you can remember the diagram very clearly, then this type of question is very easy to remember and understood. You have to know the sequence of the events, what is happening over there. Next. Sorry. Anyone has gone through this particular chapter except Anaga, of course. Morphology of flowering plants. This is from that particular chapter. Option A. Okay. Option A is the correct one. So from this is from the chapter of the 11th standard, morphology of flowering plants. So they have discussed about means firstly the symbols are given. So sometimes they ask you about symbols. This is among that particular chapter. The floral formula is one of the favorite question for the in the previous papers. So try to remember symbols, and they have described, they have discussed about four different kinds of classes, means four classic examples has given. So if possible, go through those examples, means I will recommend for others, please go through those examples. So you have to know the meaning of each stage. So zygomorphic flower, those zygomorphic means flower which is bilaterally symmetrical, means only in one direction. If we cut it, then we can get mirror images. That is what the bilateral flower, which is the characteristic of the zygomorphic flower. Then diadelphous. Diadelphous means the androgymes, the stamen part is partially joined. The androgyne part is partially joined, and they are present in the bunch. So in case of the diadelphous, two bunches are there. In polyadelphous, many bunches, more than two bunches are there. Then we call it the polyadelphous. And then superior ovary, so that means the position of the ovary in the higher position. So this is a classic, the formula that they have discussed is a classic case of the Fabaceae. That is which the example of the one of the example is the pea plant. So here are the symbols. So please go through the symbols one more time. So the superior ovary that they have talked about. So in this particular thing, firstly you can choose about the superior ovary. So if dash is present below 1, that is definitely not the case. Dash should be present below G. So which is the gynoecium, which is referred to the female reproductive part. So this is the ovary. So you can easily firstly eliminate these two things. Then amongst this, this is a sign for zygomorphic flower. So at least if you don't understand diadelphous androgyne at that time, and if you are clear with the zygomorphic flower, then you can easily select option A. So try to remember these kind of things. Means the symbols, at least go through the symbols, and if possible, the four examples that they have given for the floral formula, the Liliaceae, Solanaceae, means the potato family, then pea family. These are the classic examples that they have discussed. If possible, I will definitely recommend you all to go through those parts, because in the earlier parts, this is the one of the many times asked questions on this particular region. Okay.